Hi, my name is Kurt, and today I'll be doing a presentation about bioluminescence. So bioluminescence is a chemical reaction which produces light inside an organism. There's a wide variety of bioluminescent species, including jellyfish, fish, fireflies, glowworms, and phytoplankton. Organisms use bioluminescence for a variety of purposes, including hunting, like an anglerfish, which uses a bioluminescent appendage as bait to catch fish, phytoplankton, who use bioluminescence for defense. The predators of the phytoplankton are wary of eating them, as it would cause them to start glowing and attract larger predators, putting the phytoplankton's predators at risk. Or in communication, like in the case of adult fireflies, which use bioluminescent communication in courtship and as a way to attract mates. All bioluminescent species convert chemical energy into light. The light results from the oxidation of an organic substrate, a luciferin, catalyzed by an enzyme called a luciferase. While the specific biochemistries of bioluminescence are diverse, all include an enzyme-mediated reaction between molecular oxygen and an organic substrate. It is likely too that all bioluminescence processes involve the formation and breakdown of a four-member ring peroxide or a linear hydroperoxide. An overview of the chemical and mechanistic aspects of a major bioluminescence process, that of the bioluminescent firefly, will be presented here. Fireflies express a protein called luciferase, which catalyzes a light-producing reaction. The reaction of luciferase takes place in two general steps. First is the adenylation of luciferin using ATP, and second is an oxidative decarboxylation of the luciferyl adenylate to form an electronically excited oxyluciferin. When the excited oxyluciferin moves back to the ground state, energy is released in the form of light. The bioluminescence light emission pathway is very efficient at producing light and does not result in any production of heat. Based on mutational studies of the luciferases and enzymes in the related acyl adenylate forming superfamily, a possible mechanism for the luciferase catalyzed formation of adenylates in step 1 is shown here. So here we can see the active site of the enzyme. So all of these dashed lines right here, these are holding the D-luciferin in place. And all of the dashed lines this way are H bonds that are holding the ATP in place. And residues S199 and K206 over here are highly conserved throughout the acyl adenylate forming superfamily, and they're shown chelating the beta and gamma phosphate portion of ATP. And the key residue in the catalytic process, mainly responsible for lowering the energy of the transition state, is K529, shown right here, which is probably assisted by T343. The side chain ammonium group of K529 is shown orienting the D-luciferin and ATP for a productive bimolecular reaction. The curved arrows show a carboxylate ion oxygen atom of D-luciferin carrying out a nucleophilic attack at the phosphorus atom of the alpha phosphate of ATP. A pentavalent transition state is formed that is likely stabilized by electrostatic interactions with an ammonium ion of K529 and H-bonding interactions with a side chain hydroxyl group of T343. Protein stabilization of the transition state accounts for the catalysis of the adenylation reaction. In addition, signature sequence residues S199 and K206 function to remove the inorganic phosphate leaving group as the product adenylate is formed. The exact mechanism of the full bioluminescence reactions is not known, but the generally accepted mechanism goes as shown. Luciferase first binds luciferin and ATP in step A. Magnesium is needed to shield the negative charges on ATP. The luciferin acts as a nucleophile and attacks ATP to form a luciferyl adenylate still bound to the enzyme. Pyrophosphate is released. Following the formation of the enzyme-bound luciferyl adenylate, step B takes place and a proton is abstracted from the C4 carbon of the adenylate by a basic side chain amino acid residue of luciferase. This forms a carbanion which can attack molecular oxygen. Next, in step C, molecular oxygen adds to the newly formed anion and an electronically excited state oxyluciferin molecule is created. In step D, a ring is formed and AMP is released. This results in a high energy and highly reactive dioxetinone intermediate. In step E, the ring spontaneously breaks and we are left with electronically excited oxyluciferin and carbon dioxide. When oxyluciferin returns to the ground state, energy is released in the form of a photon of light. According to the original mechanism based predominantly on chemiluminescence model studies, red light emission, which is observed at pH 6, results from the keto form of the emitter. At pH 8, the familiar yellow-green light emission is produced from the enolate dianion form of the excited state oxyluciferin by a presumed enzymatic assisted tautomerization as shown in step F. However, more recent experimental evidence with a firefly luciferin analog is consistent with the keto form of oxyluciferin alone being capable of producing all of the colors of firefly bioluminescence. In nature, beetle luciferases display various colors of light from green to red. Possibly, luciferase modulates emission color by altering the resonance-based charge delocalization of the excited state as shown in equation 5. There are excellent summaries of the mechanistic proposals on bioluminescence color determination. However, a generally accepted consensus remains elusive. A relatively large amount of excitation energy is required to produce visible light, on the order of 40 to 70 kcal per mole. The key dioxetinone intermediate shown in the reaction sequence here contains both a strained four-member ring and a weak oxygen-to-oxygen peroxide bond. The cleavage of the high-energy dioxetinone ring is capable of releasing sufficient energy as a result of the low energy of activation required to cleave the peroxide bond, and the relief of the ring strain inherent to the structure. In the firefly, the energy released is very efficiently directed into the production of an electronically excited state of the bioluminescence product oxyluciferin. Subsequent rapid relaxation of the excited state to the ground state is then accompanied by the emission of a photon of light as previously stated. One detailed mechanistic view of this process is termed the CIEEL mechanism, which is shown here. In firefly bioluminescence, intramolecular electron transfer from the heterocyclic portion of the molecule to the dioxetinone produces a radical ion pair and the radical anion of carbon dioxide. Next, the back transfer of an electron from the radical anion of carbon dioxide to the radical form of oxyluciferin results in the formation of electronically excited oxyluciferin and carbon dioxide. So this is the structure of luciferase, and it's composed of a single polypeptide chain with residues 4 through 436 making up the large N-terminal domain, and residues 440 through 544 making up the smaller C-terminal domain. The N-terminal domain contains a beta barrel and a 5-layer alpha-beta-alpha-beta-alpha structure. The C-terminal domain has 5 beta strands and 3 alpha helices. A disordered loop connects the two domains. Scientists are not entirely sure how the structure of luciferase allows it to function. They compared the structure to enzymes that catalyze similar reactions like fatty acid coenzyme A ligase. Certain residues seem to be conserved on parts of the N and C terminal domains. This suggests that the active center is formed when the two domains come together upon substrate binding. This crystal structure of Japanese firefly luciferase is complex with a high energy intermediate analog. You can see how the two domains came together around the substrate. Scientists have used luciferase, the enzyme which is involved in bioluminescence, for biochemistry technology and lab techniques. An example of this is pyrosequencing, which is a method of DNA sequencing. Pyrosequencing can be used to determine the order of nucleotides in DNA. This type of DNA sequencing is based on the sequencing by synthesis principle, in which the sequencing is performed by detecting the nucleotide incorporated by a DNA polymerase. Pyrosequencing relies on light detection based on a chain reaction when pyrophosphate is released, hence the name pyrosequencing. Sequencing by synthesis involves taking a single strand of DNA to be sequenced and then synthesizing its complementary strand enzymatically. The pyrosequencing method is based on detecting the activity of DNA polymerase with another chemoluminescent enzyme. Essentially, the method allows sequencing a single strand of DNA by synthesizing the complementary strand along it. one base pair at a time, and detecting which base was actually added at each step. The template DNA is immobile, and solutions of A, C, G, and T nucleotides are sequentially added and removed from the reaction. Light is produced only when the nucleotide solution complements the first unpaired base of the template. The sequence of solutions which produces chemiluminescent signals allows the determination of the sequence of the template. As you can see in this chart, PPI is only released when the correct nucleotide is added to the strand, and only then can the PPI react with AMP, which forms ATP and goes on to react with the luciferase and produce light. So that was my biochemistry presentation, and I hope you enjoyed it.